Welcome to the future of understanding. This is NeuroMind Insights, where we unlock the power of your mind.
Observe how a signal moves through the nervous system. An electrical impulse travels down a neuron at high speed, but then it comes to a dead halt. It hits a microscopic physical void between two cellular structures, the synaptic cleft. Electrical currents cannot simply jump this gap. To cross the void, the brain relies on a highly specialized chemical bridge, molecules known as neurotransmitters. This establishes the core architecture of every chemical synapse. You have the presynaptic neuron, which sends the signal, facing the postsynaptic neuron, which receives it, across the cleft. When an electrical action potential reaches the end of the presynaptic neuron, it triggers small sacs called vesicles to release chemical ligands directly into the gap. The synapse functions as a translation site, where the timing and frequency of an electrical impulse determine the specific release and duration of the chemical signal. This split-screen diagram of the cell membrane shows exactly how that chemical message is received. The postsynaptic neuron's response is strictly dictated by two distinct classes of receptors. On the left, we have ionotropic receptors. These are ligand-gated ion channels, and they serve as the fast-acting mechanisms of the nervous system. When a neurotransmitter binds to an ionotropic receptor, it immediately forces a pore to open. Ions rush across the membrane, instantly altering the cell's electrical potential. On the right, we have metabotropic receptors. Notice that these lack an immediate ion pore. Instead, they are coupled to G proteins. Binding to this receptor awakens a G protein beneath the surface, which then detaches to initiate a cascade of secondary signaling and enzyme activations inside the cell. A neurotransmitter's ultimate function, whether it triggers an immediate electrical spike or a slow, sustained intracellular change, is entirely dependent on which of these two receptors it binds to. Once that signal is delivered, the chemicals must be removed. Allowing neurotransmitters to linger in the cleft leads to overstimulation, disrupting future signals and potentially damaging the receiving cell. A small fraction of these molecules will passively diffuse out of the active synaptic cleft on their own, but the nervous system requires faster, more precise termination. One method is active enzymatic degradation. The brain deploys specific enzymes into the synapse to physically dismantle the neurotransmitters on site. The other primary method relies on transport proteins. Rather than destroying the molecules, these specialized membrane proteins capture the neurotransmitters and shuttle them right back into the presynaptic neuron to be recycled, a process called reuptake. A neurotransmitter network is defined just as heavily by how it clears signals away as it is by the receptors it activates. Acetylcholine serves as our structural baseline. It is synthesized by combining the nutrient choline with the enzyme acetylcoenzyme A. This sagittal brain map highlights the macroscopic origins of these cholinergic neurons. We find localized clusters projecting outward from the medial septal nucleus and nucleus basalis in the basal forebrain, as well as the peduncle using receptor duality. It drives rapid excitation by binding to ionotropic nicotinic receptors and dictates complex modulation via metabotropic muscarinic receptors. Its clearance protocol is highly specific. Acetylcholine relies exclusively on the enzyme acetylcholinesterase, which instantly cleaves the molecule into acetate and choline to terminate the signal. This combination of dual receptor affinity and rapid enzymatic destruction allows acetylcholine to function as both the instant trigger for muscle contractions in the body and a sustained modulatory driver of memory and arousal in the brain. Next, we look at the monoamines. These neurotransmitters share a specific chemical structure called a catechol nucleus, and they are built stepwise from specific precursor amino acids. This biochemical flowchart maps the initial synthesis pipeline. At the top node, the amino acid tyrosine is converted into L-dopa. L-dopa is then decarboxylated downward to produce dopamine. Dopamine production is heavily concentrated in the midbrain substantia nigra and ventral tegmental area. From there, it projects outward through massive anatomical routes, the mesostriatal, mesolimbic, and mesocortical pathways. These networks are highly modulatory. Dopamine acts on at least 5 distinct subtypes of G protein-coupled receptors, and its signals are terminated by dedicated dopamine transport proteins executing reuptake. But the biochemical pipeline doesn't stop there. introduced the enzyme dopamine beta-hydroxylase, and dopamine is directly catalyzed into a new neurotransmitter entirely, norepinephrine. This slight structural change requires a new origin point. Norepinephrine production shifts to the locus ceruleus in the pons and medulla, where it projects out to specialized adrenergic receptors. A single enzymatic addition to an amino acid pipeline shifts a network's function from regulating reward and physical movement to dictating global alertness and sympathetic nervous system response. This new flowchart and brain map overlay tracks a completely distinct monoamine pipeline. Here, the precursor amino acid tryptophan converts to 5-HTP before stabilizing as serotonin. The origin points for this pipeline are concentrated in the brainstem's raphe nuclei, projecting far beyond to supply serotonin throughout the entire central nervous system. The defining feature of the serotonin network is its extraordinary receptor diversity. It utilizes 7 distinct receptor families, containing 14 unique subtypes, relying almost exclusively on G-protein-coupled mechanisms. To manage this vast signaling web, the system uses a dedicated membrane protein, known as the serotonin transporter, or SERT, to sweep the active cleft clean. Attributing human mood to serotonin levels is an oversimplification. The complexity of the system lies in its vast modulatory reach across 14 different receptor targets, influencing neural states well outside of a single subjective feeling. Beyond the modulators lie the amino acid transmitters. Synthesized from the metabolic intermediate alpha-ketoglutarate, glutamate accounts for the vast majority of excitatory connections in the central nervous system. Its volume is staggering, deployed at over half of all synapses in the brain. Glutamate drives this excitatory load by binding to rapid ionotropic AMPA and kainate receptors to instantly depolarize receiving cells. It also binds to structurally unique NMDA receptors. The specific ion flow of NMDA receptors directly triggers synaptic plasticity, making glutamate a mechanical requirement for learning and memory formation. This cyclical diagram details how the brain survives that excitatory load. Specialized excitatory amino acid transporters, or EATs, do not pull glutamate back into the sending neuron. They sweep it sideways into neighboring glial cells. Inside the glial cell, the enzyme glutamine synthetase neutralizes the molecule, converting it into glutamine. Only then is it safely transported back into the presynaptic terminal to be converted back into glutamate. The immense volume and excitatory danger of glutamate demand this multicellular enzymatic recycling loop simply to prevent the entire nervous system from overloading. The necessary counterweight to that system is GABA, the brain's primary inhibitory brake. GABA is synthesized directly from excitatory glutamate via the enzyme glutamic acid decarboxylase. GABA halts neural firing through a dual receptor approach. When it binds to the ionotropic GABA-A receptor, it opens channels that flood the cell with negative chloride ions, suppressing the membrane's electrical potential. When it binds to the metabotropic GABA-B receptor, it triggers a different mechanism, forcing positive potassium ions to exit the cell, achieving the same suppressive effect. This precise dual ion tuning of chloride influx and potassium efflux makes GABA the ultimate regulatory force, capable of shutting down action potentials anywhere in the nervous system. Pulling back from the microscopic cleft, this visualization of countless overlapping chemical pathways reveals the simultaneous execution of these diverse systems. Neural function cannot be mapped by tracking a single molecule. You have to trace its synthesis lineage, understand its specific clearance protocol, and map the receptors it activates. The dichotomy between immediate ionotropic action and delayed metabotropic modulation allows the brain to process a sudden physical reflex while simultaneously regulating long-term arousal and memory. This translates directly to clinical reality. Movement disorders like Parkinson's disease are direct physical failures within the anatomical projection pathways of the dopamine network. Similarly, pharmaceutical sedatives and anesthetics achieve their effects by artificially targeting and increasing the hyperpolarizing activity of GABA receptors. Deconstructing these precise biochemical pipelines reveals the brain as a collection of specific, targetable biological systems, rather than an opaque electrical web.
Thank you for watching. If you enjoyed this video, please like, comment, share, and subscribe to Neuromind Insights for more.